Mohit, if you want to go ahead and put your slides up and unmute yourself. Okay, great. So our second speaker today is Dr. Mohit Kumar Jolly. He's an assistant professor at the Center for Biosystem Science and Engineering at the Indian Institute of Science. His lab focuses on decoding the mechanisms and implications of non-genetic heterogeneity and collective behavior in cancer metastasis and therapy resistance. They focus on mechanism-based and data-based mathematical modeling, and they work in close collaboration with experimental cancer biologists and clinicians. And so we're looking forward to hearing about some of his group's recent work. Please go ahead, the floor is yours.
Thanks so much, Jen and Adam, for the exciting series that you guys have put together and for the opportunity to present some of our work today. So today I'll be sharing the mathematical models that we have been developing at different scales to investigate the dynamics of collective cell migration. And I think having Rashmi's talk before was beautiful, because some of the aspects about lateral inhibition and NOT signaling you'll also see here, although in a slightly different context. So we focus on trying to understand the dynamics of clustered cell migration or collective cell migration during the process of metastasis. There have been these reports for quite some time now suggesting that when cancer cells tend to leave the primary tumor as a unit, as a cohort, they form much more metastasis as compared to when they leave the primary tumor as single cells. CTC here stands for circulating tumor cells. People have also quantified the size distribution of these clusters, and they usually peak around 5 to 8 cells. Of course, their distribution can have very long tails as well. Clinically, these have been reported to associate with much worse patient survival as compared to single CTCs, and they have been known to be possibly formed before entering the circulation as well. So the aspects that we have been looking into ask questions at a biochemical signaling perspective predominantly, although I will go into some biophysical modeling later, both looking at what's going on within a cell, that it is capable of moving in a collective migration or a clustered migration where it has some physical contact with its neighbors, and how do such cells come together to be able to form that cluster and eventually evict the tumor. And the approach that you will see today is sort of going back and forth between developing mechanistic mathematical models and generating new experimental data based on predictions of the model, and of course generating new models based on new experimental observations. At the outset, let me acknowledge the fantastic set of colleagues I've had the opportunity to collaborate and work on. I'll be presenting some of these aspects today, focusing on this iterative cycle of modeling and experiments. So when we started looking into this in the field of metastasis, the idea of EMT was prevalent in the context that if you look at carcinoma metastasis, there are these epithelial cells which tend to undergo a phenomena similar to what is known in developmental biology in the context of EMT, where these cells begin to gain some properties of invasive and migratory traits, which usually enable them to exit the primary tumor. And once they reach this secondary site, they usually revert the properties undergoing the MET transition or mesenchymal to epithelial transition. So what was reported then in the literature suggested EMT or MET as a very binary process where cells were either posited as being epithelial, where they have very tight cell-cell adhesion with little ability to move, or individual cell migration, which was mapped onto a mesenchymal trait. There were reports about some metastable phenotypes in between, potentially some hybrid epithelial mesenchymal kind of states, but it was never usually thought about as a terminal state in itself, which can enable migration. The terminal state was usually thought of as a mesenchymal state. So that is where we began. We sort of looked at the literature which was available for EMT in terms of what different transcription factors and microRNAs were known to either inhibit EMT or activate EMT, and said, can we write down a mathematical model for the dynamics that is ongoing as a result of the interactions which are reported in the literature, and see whether this small network itself can enable more than two states, and can we have a stable state in between, which is neither completely epithelial nor completely mesenchymal, but sitting somewhere in between. And of course, EMT is much more complicated than that. So this is a first approximation that we took here. So here you see these two transcription factors. Many of you may be aware of these, which tend to drive a more mesenchymal phenotype. And there are these two microRNAs, which tend to drive a more epithelial phenotype. And what is seen in between them is some sort of a toggle switch, some sort of a tug of war that is going on. Each of them is trying to pull down the other one's levels. And therefore, usually these kinds of networks lead to two states where either one side wins or the other side wins. And that is what often leads to such exclusive phenotypes, which are seen in developmental decision making a whole lot. Each arrow or bar here represents some input output quantitative relationship. I'll be happy to talk about it later, but currently I'll not go into details. We basically convert these interactions into a set of coupled ordinary differential equations. So what did our model predict? Long story short, if you look at the x-axis here as some sort of an EMT inducer and the y-axis as the extent of EMT-ness, what you see here is that there are these three stable states shown by solid blue lines, and the dotted red lines show unstable states. We only will talk about the stable states here. So clearly there is an epithelial state with very low levels of Zeb, a mesenchymal state with very high levels of Zeb, but in between you also observe this hybrid epithelial mesenchymal states with Zeb levels lower than that of mesenchymal but higher than that of epithelial. So this is what we approximated as a state which has the ability to adhere as well as migrate, and therefore show some intermediate behavior to those of epithelial and mesenchymal, and potentially also capable of giving rise to a collective cell migration. So that's one prediction. The other predictions of this model were that now, if you look at this graph vertically upwards, if you draw a straight line here, in this region, you would cross only one solid blue line, which corresponds to epithelial. In this region, you would cross only one solid blue curve, which corresponds to mesenchymal. But there are regions in which you would have more than one phenotypes. What does that mean is that there will be parameter regions in which, even if you take a isogenic population, you can see cells in different phenotypes. These are clonal populations, but because of the inherent stochasticity in this network and multi-stability in this network, the cells can exhibit different phenotypes, and they can also switch back and forth spontaneously without necessarily needing an external signal per se. So these were the three different sorts of predictions that we made at that point, and over the years, these predictions have been validated in multiple different cancer cell lines. So here is a lung cancer cell line, for instance, stained for E-cadherin in green and vimentin in red. And what you see is that individual cells do express these two proteins simultaneously, and they continue to do so over multiple passages, suggesting it to be a stable hybrid phenotype. We also see this kind of heterogeneity in this FACS profiling, irrespective of which cancer cell line that you pick up. You do see this heterogeneity exists, so suggesting that this is a more common feature of networks that underlie these cell fate decisions. And if you were to segregate these cells and culture them independently, this is an experiment showing that they have the ability to switch back to a distribution which is heterogeneous rather than the homogeneous population that they can be. Of course, the rates of transition can depend on various factors, and I'll be happy to talk about it later. So that was one aspect, that yes, a hybrid state can exist. But what you see in this diagram, which is the same one that I showed you earlier, that the hybrid state does not exist by its own. Epithelial state does exist by its own, mesenchymal state exists by its own, but hybrid is always coexisting with others. So that led me back to developmental biology and see, again, taking the approximation of hybrid state and collective cell migration as synonymous, what is known to regulate collective cell migration. And I'm just showing one example here of graining head-like 2, which is known to form a double negative feedback loop with Zeb. Now, once you simulate this network, you do see that this diagram changes, and now you have a region in which the only phenotype that is available is hybrid. So that suggested that this particular protein, this particular transcription factor, can stabilize a hybrid state. The hybrid state existed earlier, but it is stabilized by the presence of this. So what's the validation of this? That you go back to the experiments and delete this particular protein or a similar other protein that we were able to find, and then see what happens to its behavior. So this is H1975, the same cells I showed you on the previous slide. What you see is that the migration does get disrupted. You begin to see single cells sort of flying out instead of these collective migration forming finger-like projections and protrusions, which was seen in the control cells. And these cells lose their E-cadherin and they basically only show Vimentin, which means that these cells have undergone a complete EMT instead of just staying in a hybrid state. So this particular molecule is acting as a brake on and preventing the cells from undergoing a complete EMT. We found another example. This is NRF2, which again, some of you may be aware of, is very well known to be involved in antioxidant response. And this is the circuit based on literature that we were able to curate. So what we saw is that in case of NRF2, you should see a larger proportion of hybrid cells. So again, we did similar experiments in the same cell line. We did observe that in presence of, when you knock down NRF2, you do see a disruption in migration, and we also saw a disruption in the transfer assay. Here, we did other set of experiments as well, because there is a pharmacological activator of NRF2 that is available, SFN, sulforaphane. So when you activate that, and these are bladder cancer cells now, what you see is, interestingly, that there is an increase in both E-cadherin as well as ZEB1. E-cadherin is an epithelial player and ZEB1 is a mesenchymal player. If you look at the merged image, you do see that this increase is actually quite synchronized, and individual cells have this increase rather than some cells increasing their E-cadherin and some cells increasing their ZEB1. So suggesting that, yes, if you overexpress this, you can actually increase the percentage of individual hybrid cells, which is what the model had predicted. The other aspect which we looked into is that, well, how is this hybrid EM status connected to this property of stemness, which is reported in the cancer metastasis literature, the ability to form tumors. And we did predict, based on similar mechanistic-based modelings, is that the hybrid state, again, is poised in such a way that it is able to form more tumors. And this prediction was also validated in multiple different papers. This is a Nature paper in in squamous cell carcinoma, which appeared a couple of years ago, where they showed that there are multiple states of EMT. They can all switch back and forth, and the ones that form most metastatic are the ones which are somewhere in between and not completely epithelial or not completely mesenchymal. So what has happened over the years is the idea of EMT has changed from a very binary process to a process which has multiple states in between, and cells can go back and forth. And these states they're not only existing, but their existence is apparently essential for cells to form tumors. So that's just what is going on within a cell. Next, we wanted to see how are cells coordinating their state. So if you have a hybrid cell somewhere here and a tumor and another hybrid cell located somewhere far away, how do they actually get together to form a cluster of circulating tumor cell and move? So one of the hypotheses was that they just end up right next to each other by chance. And the other is that one cell is able to induce a similar behavior in its neighboring cell and see whether it switches its phenotypes. So then we went on from these intracellular models to integrate components of cell signaling, in this particular case, NOT signaling. So again, as Rashmi introduced, NOT signaling has these different players. Delta and Jagged are its ligands, and delta is involved in lateral inhibition, and Jagged is involved in lateral induction, which is a completely different behavior than lateral inhibition. This is the circuit. Again, you can write down a set of coupled differential equations based on this, put this circuit in a lattice now, where every cell is interacting with 6 of its neighbors, if you're thinking about a hexagonal lattice. What you can see here is that NOT signaling activates EMT, and all these microRNAs inhibit NOT. So what we did was we simulated these 2 different scenarios, one in which the production rate of delta was much higher than that of Jagged, the other where the production rate of Jagged was higher than that of delta, in this lattice of 50 cross 50 cells, each of which has that circuit put in. So what you see, if you look at some patterns of spatial patterns of epithelial, hybrid, and mesenchymal cells, you do see that particularly in the presence of higher Jagged, these hybrid cells end up right next to each other as compared to what you see in case of higher delta, suggesting that Jagged might be stabilizing cells or might be enabling one cell to induce a hybrid phenotype in its neighboring cell, which is what the idea of lateral induction is. We did look at experimental data from clusters of circulating tumor cells versus single circulating tumor cells, and we did see that Jagged was actually higher in clusters, and the cells which had high levels of Jagged had high levels of both epithelial as well as mesenchymal markers, suggesting that there might be an overlap of cells with high Jagged as well as the cells which are hybrid in nature. But that's just association. We wanted to sort of take the next step and modulate the levels of jagged and then see how does it affect tumor formation. So we took inflammatory breast cancer cells, some 149, and deleted jagged in those cells with no effect on viability or proliferative ability, but you do see a huge impact in the ability to form tumors. And jagged was also seen to be one of the top 4 genes most correlated with the keratin 14, which is known to be a marker for collective cell migration, as reported by Andy Ewold's group a couple of years ago. So it was all sort of coming together. So if you look at this part of the story, we sort of looked at what is going on within a cell, identify stability factors, what's going on in cell-cell communication, and how these factors can contribute to those things. So what are the open questions which we are trying to address now? So first of all, defining EMT is becoming more and more tricky by the day, because you can define EMT based on morphology, you can define EMT based on molecular signatures, and of course based on functional traits. And we are not the only ones thinking about it. There's some ideas similar to that were put together by Andy Ewold a couple of years ago. So we need to be precise in what exactly is necessary and sufficient to define EMT. Then which of those properties which you define as EMT are actually required for collective cell migration? Again, a beautiful paper by Jen, which appeared last year, but talking about how EMT or partial EMT is similar or different than the jamming to unjamming transition, which is also reported in collective cell migration. And finally, can we question our approximation that we have been talking about of hybrid EM cells being responsible for collective cell migration, and sort of look deeper into what exactly is going on in terms of spatial temporal dynamics? And the reason I'm raising all these questions is because the way the literature is growing in the context of partial EMT states, I'm afraid it becomes an unfalsifiable hypothesis where either if you have the morphology box checked, but the molecular box is not checked when it comes to defining EMT, that's also partial EMT, or vice versa. Are both of these phenotypes equally eligible to be called partial EMT, or do we need to do a better job in quantifying this and characterizing this? So while we were thinking about this, we came across these two beautiful papers by Park and Wong's group, bioengineering there, has been developing biosensors to track the real-time levels of various different proteins involved during the dynamics of collective cell migration. So NRF2 work that I showed you earlier was actually done together with Park's group. Federico led that work together with Sam. So next we wanted to ask, we have looked at all of these three independently, NRF2, Notch, and EMT. How do they actually crosstalk with each other when it comes to collective cell migration? So what these set of experiments which were done in the same bladder cancer cell lines where NRF2 was earlier pharmacologically overactivated, what you see here is that if you either deplete NRF2 using CRISPR-Cas9 or you overexpress NRF2, in both the cases, the ability of cells to undergo collective migration decreases. So somehow the optimal levels of NRF2 are the ones which are forming most protrusions and also show the migration rate, which was a bit surprising to us. So then we started looking into how is NRF2 actually affecting EMT players. So we looked at ZEB1 and E-cadherin. These are immunochemistry images in the context of a scratch assay here. So what you observe, and again sort of quantify these things, is that and you can also look at the spatial dynamics of these different players, is that if you overexpress NRF2 via SFN, you clearly see an increase, and here is a decrease in the knockout cell line. E-cadherin increases when you overexpress NRF2, and so does ZEB1. So this is similar to the result I showed you earlier. But if you deplete NRF2, you see an increase only in ZEB1 and a decrease in E-cadherin. So if you deplete NRF2, you see cells moving towards more mesenchymal state, where mesenchymal proteins are high and epithelial are low. But if you overexpress, you see cells moving towards a more hybrid phenotype. So now you have a knob in which you can move cells from mesenchymal to hybrid or vice versa. Again, what you see here is that at the invasive edge, the ZEB levels are higher, E-cadherin levels are lower, suggesting that yes, these cells somewhere in the front at the edge are the most mesenchymal ones. And as you go deeper in the layer, the EMT-ness decreases. Now, interestingly, what we found was that if you take the intensity of ZEB1 and E-cadherin and take a product, that peaks somewhere close to 5 to 7 layers. And a product of that, again, sort of we are using as a proxy for hybrid. So what we're saying is that there are mesenchymal cells at the very front. There are hybrid cells, not at the exact leading edge, but maybe a few layers inside. And then as you keep going inside, the hybrid nature also decays and the cells become typically more and more epithelial in nature. NRF2 also was shown to change the levels of jagged notch in delta 4. So again, what you see is that in the leading cells, the levels of delta is high, the levels of notch is low, which is again lateral inhibition that is going on. But interestingly, these cells do have some levels of jagged as well. So it's a neat balance of lateral inhibition and lateral induction that is being maintained. And if you overexpress NRF2, you see that jagged 1 levels decrease. If you knock down NRF2, you see that jagged 1 levels increase. So jagged is somehow inversely correlated with NRF2 here. And you do see these gradients of all these 3 layers of notch signaling continuously. So this is when we actually had those biosensors put in, and we wanted to measure the levels of MIR200, which is an epithelial inducer in this case. So again, what we see is that as you go inside the layer, the epithelial nature increases. So therefore, you see an increase in MIR200 levels. And in the presence of NRF2, delta levels were reduced and notch levels were higher. And similar results were seen for live imaging as well. Now, finally, coming to leader cell formation. Again, what we see is leader cell formation is maximum when NRF2 levels are optimal. If you decrease it or if you overexpress it, you see that leader formation is decreased. So then we closely looked into what is going on at the leader cell edge in this particular case. So what you see is that the leader cells, again, in this case, peak adherin is low, ZEB1 is high, jagged is high, as well as delta is high. So this is not a completely lateral induction. It's a good, neat balance of lateral induction and lateral, it's not completely lateral inhibition. It's a good balance of inhibition and induction that is being maintained here. And the cells which you see, that these leader cells, they are usually more mesenchymal, and they tend to have high delta, but with the presence of jagged and also low notch. So there is lateral inhibition which is going on, making sure that there are only a limited number of leader cells and not everyone becomes a leader cell. But there is potentially the presence of jagged somehow, acts, that's what we think currently, acts as a buffer so that it ensures that in case one of the leader cells is perhaps not being formed properly, that there are lateral induction-based mechanisms which somehow tend to compensate for that. So now these were all different observations which we then put together in this mechanistic model, where you have the circuits that I've been showing you earlier, an RF2 circuit, notch circuit, and there is this invasive edge. So there is a constantly, as you go inside, there is a decreasing levels of this EMT inducer which is present. So based on that, we were actually able to recapitulate most of the observations that I talked to you about, that there are initially much more mesenchymal cells, but as soon as you go inside, there are these more hybrid cells and epithelial cells eventually, and this crossover where hybrid cells is maximum, the higher the levels of NRF2, the earlier to the invasive edge this crossover is. We did further sort of set of simulations where we looked only at the first layer, only at the invasive edge, and did simulations where you can change the levels of NRF2 production rate. So this is like overexpression of NRF2 happening as you move from left to right in each of these scenarios. So again, what you see is that as these levels increase, the MIR200 levels increase, which means cells are becoming more hybrid in nature because the notch levels are also constantly increasing. And you do see cells which are both delta positive as well as jagged positive. So two ligands which are usually known to do completely different things, you can see hybrid cells here as well. So I'll just summarize here that a EMT is not just a binary process. You can have cells which are maintained stably for longer durations in one or more of these hybrid states, which can be facilitated by some of these intracellular as well as intracellular molecules. We do need a much better characterization of these hybrid states before it runs into being an unfalsifiable hypothesis. And we do need better characterization of how are these states switching back and forth during the process of migration itself. What is the time scale that is involved there, and how does that support the dynamics of collective cell migration? And eventually, we need to do a much better job at integrating some of the biophysical aspects of cell adhesion, cell matrix adhesion, etc., and the underlying biochemical. And there's clearly a feedback loop that goes on in both directions. So these are not as segregated as presented here. And together with these multi-scale mathematical models, which present a beautiful opportunity to integrate these two different aspects and also go back and forth together with the experiment. So what's the ongoing work currently in the lab? We are, of course, looking into the more biochemical, intracellular and intracellular signaling dynamics of how are hybrid cells maintained during collective cell migration. We're also looking into more biophysical models where we don't worry about the molecules per se to begin with, but talk about different forces that cells apply on one another as well as on the matrix, and how that affects the dynamics of collective cell migration. There's a beautiful paper by Ushasi and Andrew Mugler. Ushasi is now a postdoc in the lab, which is very consistent with what we have been saying. It's a completely biophysical model talking about how intermediate adhesion, cell-cell adhesion, which we would map onto a hybrid behavior, actually maximizes the migration velocity of multicellular clusters. And the direction which we are beginning to develop more recently is trying to integrate these two aspects. There was a recent study we did where we were able to connect how EMT affects the stiffness of ECM and how ECM stiffness again induces EMT. So those are the kinds of directions in which we are moving on currently. With that, I would like to thank you all for your attention, and we'll be happy to take questions.
Fantastic. I really like your final slide there. That's great. So we have a couple of questions coming into the chat, and folks, please continue to drop them there. Use the raise hand function and we'll call on you. So from Ian Wong, he says that he agrees about the difficulty of testing EMT without clear definitions, and he wonders if it would be simpler to investigate these pro-migratory phenotypes and collective behaviors in an EMT agnostic framework.
Definitely, I completely agree with you, Ian. I think we should not get stuck in the nomenclature, and we should sort of try to identify what the functional aspects are necessary and sufficient to. And there can be more than one functional programs, or there can be more than one molecular program sort of mapping onto the similar functional traits. So I completely agree with you.
I guess, and just a quick follow-up from me on that. There was the paper that came out, I don't know, a year or 2 years ago. That was the big sort of consortium effort with like 50 authors, everybody, the consensus view of EMT and partial EMT. Do you think that fits in with what you're thinking, or is that still too much of just like, there's so many, even within that framework, there's so many different ways to define a hybrid state or a partial EMT?
I think that paper is a step in the right direction, but we need to move ahead. And of course, there is context-dependent reasons which sort of come in. Even if we don't cross the boundaries of development and cancer, different cancers also exhibits like the different molecular profiles and so on. So we need more. While such reviews and perspectives are great, but we do need more research activity on that front also to sort of characterize these traits better.
Yeah, for sure. I mean, one thing that I found, when writing my paper is that so many of the references and the framework that we built, we end up just referring to reviews and reviews and reviews going back in time. And then when you actually dig some of the claims that we're all making over and over, there isn't that much actual experimental data, right?
I completely agree. Yes.
Anyway, perfect.
The next question is from Prabhu. I don't really understand it, but I'm going to interpret it the way I get it, which is how do the cells go between these, EM hybrid versus E versus M states as the cell is reaching its target? So I guess once it's trying to finish migrating. Have you thought about that?
So these are dynamic processes in the sense that the cell which is epithelial at time t1 may not be epithelial at time t2, and this switch can happen, of course, due to the influence of its neighbors, but it can also happen because of stochastic fluctuations in that individual cell itself. So we need to understand these different mechanisms which are capable of making this switch, which operate at different time scales and length scales. And whether this so-called steady state we are thinking about is reached at an intracellular level, or is this reached at a tissue level where there is some sort of coordination across? I don't know if I understood the question correctly, but I'll be happy to discuss further.
Prabhu, did you want to follow up at all? I saw you were unmuted.
Hi, yeah. So I was just wondering if as the cell moves, does it mean that when it is trying to migrate and it's trying to invade some other space, does it is this EM and EMT states are like this happens during the transition, which means that it's initially epithelial and when it wants to move, it's mesenchymal. And I mean, during that moment, if it wants to settle somewhere, does it mean that it just tries to become epithelial again because it wants to stay there? So I was just wondering if this is what happens in systems like wound healing, where the cell actually has a target. So I was wondering if this state is achieved to reach a target, to settle somewhere. So I was just seeing if this transition is what happens then.
I think that's an excellent question, Prabhu. Yes, no, now I got it. So the last part that I showed you, that was all in wound healing scenarios. I mean, in scratch access scenarios, which are trying to mimic wound healing. Okay. So there we are trying to exactly understand how are these changes happening, and that's why we have these live cell imaging biosensors in addition to immunochemistry to sort of see how those changes are happening. And in this case, it's just scratch assay. But if you were to make more realistic assays for wound healing by adding other kind of immune cells, these the technologies that we are trying to build through collaboratively by integrating imaging, mathematical modeling, and these different perturbations is a step in that direction.
Perfect. Thanks.
Thanks.
And we have a question from AJ asking you to explain a bit more about the role of snail in maintaining the hybrid state.
So snail, the way we think about it, it's like a rheostat in some sense. So depending on where you are at the snail levels, you can be locked in a completely epithelial state. If the snail levels are relatively very high, it's in a mesenchymal state. But in between, there is a window where all the cells in a population can have the exact same snail level but different phenotypes. And the reason that is happening is because that network is capable of giving rise to multiple phenotypes. The genotype to phenotype mapping is not one-to-one. On the other hand, work I did not present today, we have dug deeper and distinguished between snail and slug, because slug was consistently reported to, again, going back to even mammary morphogenesis literature by Keith Mostow I think almost 15 years ago now, suggesting that the slug has a more potent role. And we see the same thing. If you sort of now look at snail and slug differently, which we did not in the initial attempts, we see that slug particularly stabilize a hybrid state, and snail does not. Snail pushes the cells all the way to more mesenchymal, but slug maintains them in between.
Right. I really, really enjoyed your talk. And actually, I think this framework that you have described really is extraordinarily useful for some of the work we are doing in the lateral line primordium for answering and thinking about a lot of unresolved questions. So thanks a lot.
Thank you so much. I'll be happy to chat further. Thank you.
Yeah, I'll actually get in touch with you again.
Sure.
Okay, fantastic. And then I guess I just want to maybe as the last question, get your thoughts on something that's always kind of I don't know, I guess it's kind of bothered me a bit, which is that a lot of these in vitro studies, we're using these, sort of epithelial-ish cell lines, but they're being cultured in, while being exposed to serum, which has TGF-beta in it. And so I guess I'm wondering, how do we go from thinking in vitro, which specifically the serum and TGF-beta thing is a thing that always sort of sticks with me. How do we then fit that? I mean, because presumably we're looking at cells that they're not truly epithelial, right? So we're never really starting from an epithelial state and moving into a partial or a hybrid state. So yeah, which I know I'm not really framing this as a precise question, but what are your thoughts about connecting that to then the in vivo circumstances?
So I think a more recent work which has been done looking at phenotypic heterogeneity in an individual cell line itself has shown, including the FACS plots that I showed you, that there are subpopulations in that. And the other thing we have done is we have, using sort of bioinformatics methods, we have tried to quantify the extent of EMT in a microarray or RNA-seq kind of profile. So we say took MCF7, the beloved epithelial cell line, and we took different data sets, all on PubMed, and tried to quantify, are all of them really epithelial? They were not. Mm-hmm. So there was varying degrees of mesenchymal program which was there, which could happen because there are subpopulations and they get enriched due to various culture conditions, including serum, for instance, or they get selected due to other pressures. So I agree with you that I think in vitro scenarios have their limitations. So I think tracking these using live imaging kind of scenarios, moving to 3D, for instance, that's the first good step, moving towards organoids, which Andy World and others have been doing for quite some time now. Anything to do with dynamics, it would be fantastic, not just static. I mean, earlier when we started looking into EMT literature, it was day 0 and day 70 of Vera treatment with no points in between 10 years ago. So that we need to graduate beyond that.
Absolutely. I mean, yeah, right. The field has grown enormously in the past decade, but I really, really like your point about thinking about what does heterogeneity actually mean, right? Because it can mean a given cell with a hybrid phenotype, or it can mean multiple cells of different, phenotype, different gene expression profiles all mixed together. And a lot of our assays don't distinguish between these at all, exactly. Right? And so unless we're thinking about thinking about distinguishing those, it's really hard. Yeah. All right. That's I really appreciate both of your talks. Rashmi, thank you so much for kicking us off. That was fantastic. And Mohit, that was obviously wonderful as well. And I think we'll wrap up now. So I'll end the live stream and just thank you both for your talks, and we'll see you all next